Hi, my name is Derek Reiman, and I'm a PhD student at the University of Illinois at Chicago, Department of Bioengineering, under Professor Yang Dai. Today I will be talking about MiMeNet, a framework that can be used to explore the microbiome-metabolome relationships using neural networks. In our work, we will be looking at microbiome data. The microbiome consists of microscopic organisms cohabitating in a shared environment, and has been shown to have a significant impact in both host development as well as the pathogenesis of various diseases. The microbial community is believed to functionally interact with their host at a metabolic level through symbiotic interactions and co-metabolism, and recent studies are beginning to highlight various metabolic dysregulations leading to the development of metabolic diseases. As paired microbiome and metabolome data become available, integrative analysis tools to uncover microbiome and metabolome interactions would be extremely useful. Being able to identify microbe-metabolite interactions, especially in terms of the development of disease, would allow us to understand better how the microbiome affects the health of the host, and in addition, the knowledge can potentially provide insight in the development of therapeutic treatments. Here I'll be describing MiMeNet, a neural network framework designed for predicting metabolomic profile for microbial features. In addition, it'll derive microbe-metabolite interactions based on the learned models, as well as provide insights for the causes of metabolic dysregulation in diseases, which could allow for future hypothesis generation. An overview of MiMeNet is shown in the bottom left of the poster. MiMeNet takes in paired microbiome-metabolome data as its input. After pre-processing and filtering, a neural network model identifies metabolites that can be well predicted from microbial features. The weights of the trained neural network are then used to construct micrometabolite interaction scores. The interaction score matrix can then be biclustered to identify microbial modules and metabolomic modules, and we can use these to construct a module-based interaction network. In this poster, we will look at how MiMeNet works on some inflammatory bowel disease data. We used paired data from the prospective registry and IBD study at Massachusetts General Hospital, which we will call PRISM. The dataset contains 123 patients with IBD, 34 healthy patients, with a total of 201 microbial features at species level and 8,848 metabolites, of which 466 were annotated. In order to identify well-predicted metabolites, we perform 10 iterations of 10-fold cross-validation, where we predict the metabolomic profile from the microbial features. We then evaluate each metabolite using the Spearman correlation coefficient. To determine the correlation cutoff, we perform a similar experiment of 100 iterations of 10-fold cross-validation, except this time we shuffle the samples in each dataset separately for each iteration. This gives us a background distribution of correlation values, and we can set a cutoff at the 95th percentile. A diagram of this approach is shown at the top of the results section. In the middle of the results section of the center column, we show a list of the top correlated metabolites identified by MiMeNet. We find that even though MiMeNet was given no information about IBD status, many of these metabolites have been associated in previous studies. We also show what the two distributions of correlation look like. Our observed correlations are shown in orange, and the background is shown in blue. The red line shows the 95th percentile cutoff, so any metabolite to the right we consider well predicted. Lastly, we show how MiMeNet's prediction compares to a recent tool called MelinPan. MelinPan uses linear elastic net regression to model individual metabolite abundances for microbial features. We notice an overall improvement when comparing MiMeNet to MelinPan, and we believe this may come from the neural network's ability to model nonlinear relationships, as well as the benefit of multitask learning to predicting the entire metabolic community. Note that we only compared the 466 annotated metabolites, but MiMeNet was trained on all 8,848. Next, we want to interpret what is going on within the model. For this, we use Olden's method of variable importance for neural networks, and it works by multiplying all the weight matrices together. For us, this results in a matrix where each row represents a microbial input feature, and each column represents a metabolite output feature. The value of a specific position in the matrix describes the influence of that microbe on that specific metabolite. We generate a single interaction matrix as the mean value of the matrices from the 10 iterations of cross-validation, and filter only the well-predicted metabolites. We can then bicluster this matrix to identify clusters of microbes and metabolites that follow similar interaction behavior. We can see what that biclustering looks like in the top left of the rightmost column. The module clustering is also annotated here. Using this clustering, we generate a bipartite graph as shown below. Here, the nodes on the left are microbial modules, and the nodes on the right are metabolomic modules. In addition, we identify disease-related modules using a Wilcoxon rank sum test, where a module's value is the average feature value of the features within the module. We see that the disease-related modules include fatty acids, bile acids, and glycerophosphocholines, all of which have been shown to be significant in IBD. However, we also see interesting non-disease-related modules, such as the green module in the top right, which we believe to represent a large group of amino acids. Lastly, we compared the predictive performance of our microbial modules compared to the original microbial features, as well as modules constructed using WGCNA, or whole genome correlation network analysis. For this, we used tenfold cross-validation using neural network models and evaluated the performance using the area under the receiver operating characteristic curve. We observed that MiMeNet's modules outperformed WGCNA's modules and had no loss in predictive performance compared to the original data. This indicates that even though MiMeNet had no information about disease status, it was able to organize microbes into meaningful modules that could still capture this disease status. In conclusion, we have developed a framework called MiMeNet, which uses paired microbiome-metabolome data to learn micrometabolite interactions in a data-driven approach. It trains neural networks to identify well-predicted metabolites from microbial data and uses the trained neural networks to construct a module-based micrometabolite interaction network. We believe MiMeNet can facilitate the identification of metabolic activity of microbes and their association to host disease. Thank you for listening, and I hope you enjoyed the presentation. If you have any questions, feel free to reach out to either myself or Professor Yang Dai. Thank you again.